Hey guys, it's Miss Bostic here. I'm going to talk to you about how to fill in Punnett squares and how to analyze the data and the information that you have once it is filled in. So, Punnett squares, these determine the probability of the traits that are passed on to offspring. And so that means that if I wanted to, let's say, breed dogs, and I have a mom dog, dad dog, the mother dog, she has brown fur, and then the dad dog has white fur. In my example here, the brown fur is going to be dominant, and so for that one, we'll say that it has the alleles of 2 capital B's. Now remember, anytime you have capital letters, that's going to represent the dominant trait. So that means that it's going to shine through. It's going to show how many. Show that trait that it stands for. The dad, he has white fur, and so for his white fur, his genotype is going to be 2 lowercase b's. Lowercase letters represent recessive traits. Recessive traits, the only way to see them is if you have 2 recessive genes together, just like the dad dog does. So let's fill this in. So we're going to fill in this Punnett square. For this, it doesn't matter where you place either parent. You just need to have one parent going along the top and one parent going along the left side of this. So I'm going to take the mom's information and I'm going to write that on the top. So 2 dominant alleles. The dad's information we'll keep in blue, but that you'll write down the side. So lowercase b, lowercase b. When it comes to filling in the Punnett square, this information here fills in all the squares to the right of it, and then this information fills in down below. So that means that, let's start with the top. So this capital B is filled into this square, and it fills into this square. So I have capital B, capital B. Same thing on the right side. Now, as far as on the left side here, this lowercase b goes here, and it goes there. Same thing on the bottom row. Alright, so that's filled in. Now when it comes to the type of questions or the things that you'll really need to be able to analyze and look at on these Punnett squares is being able to tell or calculate what percentage of the offspring would have the dominant trait, what percentage would have the recessive, or how many of the offspring are heterozygous, how many of the offspring are homozygous. So by looking at each of these squares, I've got 4 squares out of a whole, so that means that each square stands for 25%, or 1/4 of the overall outcome. So looking at this, if I need to calculate how many of my offspring would have brown fur. So for it to have brown fur, it has to have that capital B there. Looking at this, each of the 4 squares have a dominant allele in them. So that means that 100%, or 4 out of 4, would show or would carry that dominant trait. And that means that 0% would have white fur. You also need to be able to tell the homozygous and heterozygous output. Homozygous means that it's the same. So that means that it would either be both dominant or it would be both recessive. In my example up there, I actually have 0% being homozygous. Heterozygous means that it's mixed. It'll be dominant mixed with recessive. My example up there, I have 100% of them as heterozygous because each of the squares show a dominant and a recessive trait together.